Challenges in Environmental Sciences seminar series. And somebody in PI, it's sponsored by PI, somebody in PI came up with that acronym. And you'll see a number of these kind of seminars, and the idea is and the idea is that this is kind of a think tank for where the whole field of environment and environmental science is where it's going. And for us to engage kind of visionary speakers. And so turning to the speaker, I'm so delighted to have Toby Kiers here. Toby is from the University of Amsterdam, where she holds chair is called a university chair.
University
She also
chair,
has an appointment at University at Oxford in England. But turning to, and she got a PhD at Davis, and so turning to the questions, we all, one of the most challenging and remarkable things is to try to understand how species interactions influence our natural world, right? That's one of the things, it's remarkable. But one of the greatest mysteries about species interactions is when different species decide to do things together. Collaborate in like a mutualism or something like that. And it used to be that we just, we classify that as a plus or a minus, and that was kind of it. And what we're beginning to understand is that it's much, much more complicated than that. There are different strategies of a host and a symbiote that interact. And the other thing, it's not just that it's complicated and really remarkable that way. The other thing we're beginning to learn is that those interactions have tremendous leverage on how ecological systems are structured, including all the way up to whether a tropical forest can take up carbon dioxide for a long time or not. So the power of these interactions they scale up to the whole system in remarkable ways, and that's exactly why we're excited to have you here, Toby. Yeah. And I have the title of your talk is Resolving Host Microbe Conflict.
Yes.
Welcome.
Thank you.
I'm going to turn down the lights.
Great. Yeah, okay. Thank you. Great. Thank you for the nice introduction. Yeah, so I'm going to talk about conflict today, and I wanted to start with a series of images. So I want you to tell me what these images all have in common. What do these images have in common? They're black and white, right? But they all involve children. And what's interesting about these images is that they all involve children being sucked underground, right? So there was this photographer. His name was Arthur Hess, and he was a photographer in the 1960s. And what he did was he would speak with children, and he would recreate their nightmares, and then take photos of those nightmares. And so there was this, Arthur Hess realized that there was this sort of this common theme in children's nightmares about them being sucked underground, right? So why this fear? Why this fear of the underground? And I think actually, people say that we know more about space than we do about what's under our feet. And I think it's largely true. It's really unexplored territory, what happens underground. And what we do know about what happens underground is absolutely fantastic. So we see these very sophisticated networks populated by microbes that have evolved strategies to manipulate other microbes as well as their environment. And as an evolutionary biologist, it's these kinds of interactions that interest me. So our group studies the evolution of cooperation, like Lars says, mostly between different species, so mutualisms, and we study how cooperation evolves and persists. At least that's what we used to do. So recently, I realized that actually I wasn't so interested in cooperation. Instead, I became much more interested in conflict. Now, I almost think that cooperation is like the dull cousin of conflict, right? All of the action, all of the tension, it's there in the conflict and not necessarily in the harmony. So in our group, what we try to do is sort of we sort of act as rebel rousers, right? Even if you've got a relationship that has evolved in nature, we try to create conflict in that relationship and then figure out what happens. When you introduce conflict, basically it's like looking for the weak spot in an interaction. So basically, as we manipulate cooperation, we force individuals to cheat in partnerships, in these mutualisms, and then we look to see how the other partner responds. That's the basic framework of what we're doing. And what's happening in the underground is really interesting. There's this tension where microbes, and the first part of the talk is going to focus a bit on microbes, some macro stuff. But we've got these interesting strategies where, like a microbe that's growing on a healthy host, let's take just a normal host, healthy root system, where a microbe could potentially share in the collective benefits of being on this healthy host while paying none of the costs. And what that does is it creates tension, but it creates it in two different spots. And I think actually if there's one sort of thing to take home from it, it's where these two spots exist. Like, we know that there's a tension between the host and the symbiont, right? They can both keep their resources while paying as little as possible for that relationship, then they'll tend to do better. And I think a lot of the conflict is studied between host and symbiont. But where some other really interesting conflict is among the symbionts themselves, colonizing a single host. And a question that we've really been focused on in the last couple of years is, how is that conflict managed when you've got multiple symbionts on a single host? So probably the most sort of obvious solution to this problem is to evolve some sort of top-down control, where it's the host that controls the fitness of the symbiont, right? It's simple enough, it seems to work. And there's evidence that this is a very successful strategy. We see it in legumes that interact with nitrogen-fixing bacteria. If the bacteria fail to fix nitrogen, then the legumes actually suffocate those individual nodules and reduce the fitness of the rhizobia. We see it in protists that use algal symbionts. And if the algal symbionts are not producing enough carbon, even if it's just because of low light levels, then the protist can digest those symbionts. Of course, you also see it in these squid, which can expel specific bioluminescent bacteria if the bacteria fail to produce light. So these are all examples of host top-down control. Now, where we see these types of dynamics is when you've got multiple competing symbionts on a single host. Okay? We're not seeing it in situations where there's only one symbiont per host. We also see it when there's environmental variation. So as soon as you add these two components, competing symbionts and environmental variation, for instance, in the legume rhizobia symbiosis, if you have high nitrogen and the symbiont's no longer needed, this is also a way to introduce conflict into the system. So if you vary these two components, symbionts and environmental variation, this is really where we think the conflict emerges. Now, when we look at the evolution of control of hosts, one of the things that we see as an evolutionary solution to this problem of control is this idea of compartmentalization. So what do I mean by compartmentalization? Well, it's somehow that the host can put the symbionts into very distinct compartments. And what that does, it actually decreases conflict among the symbionts themselves. It's because it's isolating them. And it also potentially allows hosts to monitor the benefits of those individuals. It creates sort of a feedback between the symbionts and the hosts themselves. So compartmentalization is a really nice solution. I think these legume nodules are sort of like the gold standard of compartmentalization, where we see that legumes have evolved ways of controlling oxygen gradients, control of sugars. And what's the most amazing is that the legumes, when they interact with these bacteria, they tend to compartmentalize them so there's only one rhizobia per strain. So one individual bacteria that forms the nodule and reproduces inside that nodule. Now, there are exceptions to these cases, and those are the cases that are really interesting when you can actually get multiple competing symbionts in a single nodule. And there was some recent work done by Joel Sachs's lab that actually found that some legumes are able to target single rhizobial strains inside a single nodule. Again, is this because of some sort of compartmentalization? It's not known. But what's interesting is not all hosts have evolved this level of sophisticated control. So one of the species that we're working on right now is called Parasponia. Has anybody heard of this? It's a tropical tree, and it grows in Borneo in Indonesia. And it's the only non-legume to evolve the nitrogen-fixing symbiosis, right? So this is an incredible system. It's this group outside the legumes that can make nodules and form these relationships with rhizobia. And what's interesting about it is that it, for all this sort of interesting reasons about, well, it provides an evolutionary replicate for how these mechanisms emerge. But more interestingly, it gives us a glance at mutual evolution at an earlier stage, when all these sophisticated mechanisms of control haven't yet evolved. So when you look inside a Parasponia nodule, it's actually a mess. The rhizobia aren't in these organelle-like structures called symbiosomes. They stay in the infection thread. And in some cases, the rhizobia make a nodule and then actually start to digest the host tissue. So there's real conflicts. This is why it's an exciting system to study, because you're still on that edge of conflict. So here's just a quick, very simple graph where you've got number of nodules on the x-axis and total plant weight on the y-axis. And this is looking at the Parasponia system. And what you find is that when you've got a high fixer in blue, you've got this nice correlation, fitness correlation between the rhizobia and the plant. But as soon as you inoculate it with a lower fixer, something that's not fixing a lot of nitrogen, you can still see that the rhizobia is able to produce more nodules, increase its own fitness, but this time at the expense of the host. Now we're trying to understand, okay, so what hasn't Parasponia evolved yet that makes it so it can control the fitness of the bacteria? What is it about it that it hasn't? And it's not compartmentalization, because even though this is still a very, very early mutualism, it still has only one strain of rhizobia per nodule. And not only that, but it's a very primitive way that the rhizobia get in. They actually come in through a crack. So it's in, like, for instance, Medicago, you have this very elegant root hair that curls up, and it's this beautiful molecular dance. And in Parasponia, the rhizobia still just get in through the cracks, yet still in that way, tends to be only one rhizobia per nodule. Okay, so that's been a lot about host and microbe, but I became really interested in this idea of compartmentalization. And actually we started a project recently to look on a macro scale with other organisms, and that took us to some islands that are off of Fiji, where there is a plant, a newly discovered mutualism that involves this plant here. What you're looking at is actually a domatia of a plant, and the branches coming out are the leaves of the plant themselves. Now, surprisingly enough, this plant actually looks like a giant nodule, but the locals call it elephant testicle. That's the name of the plant in the local language. And you can see in a few pictures, it's very hairy and very large. So here I am holding an elephant testicle. And it's called squamularia. And what's interesting about it, the system was first introduced by Chominsky and Rayner in a Nature Plants paper in 2016. And what's interesting about this mutualism is it's the first example of a plant, an obligate farmed plant mutualism. So we know that ants farm fungus, for instance, but there's been no example yet of an ant actually obligately farming a plant. And so these plants, they live up in the canopy of the rainforest in Fiji, and it's only found in Fiji. And these ants, they grow these incredible cities. You can even use that word, of these plants up in the forest raintop. And neither the plant nor the ant can survive without the other. So here's a video of what it looks like. And basically, why it's a farming mutualism is that the ants, they actually propagate the seeds of this epiphyte up in the tops of the trees. They've evolved these special mechanisms whereby they dig the seed into cracks of bark. Now the plant has evolved, here you're looking at the domatia, the plant evolved these very specialized structures inside its tissue called warts. And the ant actually enters into the hole and defecates on these very, very specific structures in the plant. So not only is it propagating it, but all of the nutrients for the plant are coming from the ant. So it's also fertilizing it. And then as you can see from the video, it's also a fierce defender, right? So you just even kind of go up, and you're up in the canopy, so you're pretty high up. And if you touch one, you're just covered with ants right away. They're very, very, very aggressive. Now in return, they provide food bodies for the ants and nesting spaces, which the ants are very limited in. So here's one of the larger elephant testicles. You can see it take both of us to hold up this one. That's Guillaume Chominski, who's the one who first published on the mutualism. And you can see here's a picture where really small seedlings. And so there's a huge variation in sizes and how many each of these plants can provide housing for a number of ants. And we first became interested in it because we were interested in asking, well, how good are these farmers? How good are these ants at actually farming? So we started looking at a light gradient up in this canopy and asking, are the ants preferentially planting the epiphytes in high sunlight? And this is a big question because a lot of these rainforests are being chopped down and light levels are really changing. And so we looked at this gradient, and it was actually quite interesting that when the ant plants the plant in high light, it gets rewarded with more food bodies. And we also find that the ants, the plants themselves are better defended from herbivores, right? So the plant benefits because it's in high light, and it's better defended by the ant's getting more food bodies. But then there's less herbivores, and so more of the nitrogen actually is coming from the food bodies. So there's this contrasting benefit between high light levels, this trade-off between high light levels and less herbivory and less nutrients being provided by the ants, because they're not eating the insects anymore and using that to defecate on the plant. Another really interesting, and this is going back to compartmentalization, is that I've been talking about just the obligate system now, but there's also facultative relationships between this ant plant. And in these facultative relationships, you see this strict compartmentalization of the ant species. So the nesting space is really, really intense competition for nesting space. So there's this constant turnover. And if ants come in contact with each other, they're really going to fight to the death if they're in the same cavity. So what we see are these multi-cavity domatia, which, like in nodules, decrease the conflict and actually allow for coexistence. So what you're seeing in this picture is an X-ray photo of where each color represents a different ant species and how they're separated inside. But when we look at the obligate plants, here's another really small one, we just see a single partner. So are they moving away from this multi-species coexistence with individual chambers to a single chamber with one partner? So those are the kinds of questions that we're asking. Basically, we're trying to understand more generally when these organisms have such a low level of conflict that the two partners' interests become aligned. And when this happens, and it can happen, there's some really interesting examples of this happening in nature, then we get a new level of organism, where basically partners that originally were two different species become one. Some of the best examples are with these flatworms, for instance, that use these symbionts to harvest chemical energy. And that source has become so, the host has become so dependent that it's a really gray area now between defining what is an endosymbiont and what is an organelle. I have to go to conferences a lot, and there's actually an ongoing debate. When do you call something an organelle and when do you call it an endosymbiont? And in some of these cases, the hosts have lost their whole guts, their whole ability to digest, because they become so dependent on the symbiont. So we're interested in trying to understand what explains this diversity, right? So why is it that you have some relationships where you have lots of conflict and some where they come together in such an elegant way that they become a single organism? Can we predict what symbioses are on particular evolutionary trajectories? And is it just a matter of time and we see integration, right? There's always this sort of, even as evolutionary biologists, we know that, evolution isn't directional, but when we see this integration, we think, oh, okay, well, perhaps most organisms are on their way to integration. But, yeah.
You might have said this. Are there other examples where an ant forms a partnership with some kind of plant?
Oh, there's lots of examples where ants form partnerships with plants. Usually it's a defensive relationship, but this is a farming mutualism where they're actually propagating. So there's lots of examples of ant-plant relationships, and a lot of the work of people at this department is on those. But this is the only example where, and it's important to know what it means to farm something, right? It doesn't just mean to increase the abundance. Like a really strict definition of farming involves propagation, fertilization, and defense. Okay, so one of the big projects that we have right now is what we're calling this global symbiont database. And we're trying to get as many, it's kind of naive to think we can do this, but, if we don't, right, somebody should, to try to sort of catalog, just a first stab at symbiont relationships and asks, well, is there something that predisposes certain groups to higher symbiotic dependencies? Right? Is it something about the environment that we can say, okay, well, if we look at this environment, we sort of expect high dependencies on mutualisms? Or is it something just about the function of the symbiont themselves? Or is it some characteristics of the host that makes it so it's likely to evolve higher dependency on symbionts? So we've created this database where we look at just, examples of organisms that are forming symbiotic relationships. And right now it's mostly stuck to just hosts and microbes. So we're not at the macro level yet. So not full mutualism database, but really just about symbionts. And we have data on their transmission and their habitat and their habit, how much they move, their genome size, their function. And so recently we published a paper that just took a first stab at sort of understanding the emergent patterns that we see in this symbiont database and asking, okay, well, what is it about particular relationships, and can we then make predictions about levels of dependency based on those kinds of traits? And actually, the first results were almost a bit obvious. And maybe as we get more examples, especially from marine systems, right? There's still so little understood about marine symbioses, and that could totally change our view of these patterns. But here you're just looking at different bacterial symbionts and some of their hosts. It's more just for illustrative purposes. But what we found was that the greatest dependencies are likely to evolve on nutrient provisioning. So symbionts that provide nutrients and that are vertically transmitted. Pretty simple. I think that's sort of what we were thinking. We also found this negative correlation between host dependence and symbiont genome, and that's in vertically transmitted symbionts, but not in horizontal. So that, again, is sort of intuitive. It just means function, population structure. They're important in driving dependence. Now, I think the marine stuff might change that a bit, because there are examples where hosts evolve extreme dependence, obligate dependence on symbionts, but they're still getting them horizontally from the environment. And it's not all necessarily increased dependence. So we have some other work where we tried to get a better resolution, for instance, on some of these ancient mutualisms. And that's the second half of my talk, is looking at the mycorrhizal mutualism. So the mycorrhizal mutualism involves a fungus that's obligately dependent on a host for carbon. So it can't grow without the host. Host gives carbon. In return, the fungus gives phosphorus and nitrogen, mostly phosphorus, but other nutrients as well. And what's interesting about the symbiosis is it's the ancestral state of plants. Right? We think that this mutualism with fungus actually helped the first plants emerge from an aquatic state to a land state. So it's the ancestral state, and it's about a 450 million year old symbiosis. And it's only been lost in that evolutionary history about 20 times, 20 odd times by our recent analysis. And in those cases where it has been lost, there's always an explanation, which is quite interesting. There's only really a few cases, one or two, where we don't see it being replaced by something else. Usually we see a switch to another kind of microbial mutualist, or evolve one of these very crazy strategies like carnivory or cluster root or parasitic plants, where you evolve a very different alternative for extracting nutrients from the environment. Okay, so that's the first half of my talk, where I really wanted to sort of look at big patterns of host control and evolution of symbiotic dependence. Now, most of the work, and I think most of the work in general, in terms of host microbial relationships, it's been so host-centric, right? Everything is concentrating on the host and how the host can control the symbionts. And I think that actually what we're missing is sort of probably the more interesting side of the story. And that's about how microbes are actually manipulating the host and how they're evolving their own strategies to deal with this type of symbiotic lifestyle. Okay, so I wrote up here plant microbiome, and I think that people say the word microbiome, and now my eyes just glaze over with sort of boredom when I hear about microbiome. So it's just like, there's just, and I think, it's maybe it's some sort of weird bias, but it's also that I think so far, and this is going to change, but I think so far it's been, very, all about characterizing, and there's just hasn't been that injection of evolutionary theory into microbiome research that we need. But why I put this up is that, we keep hearing that same number about the humans are only 10% human, right, because the rest is bacteria. But for plants, it's even worse, right? Plants are only about 0.1% plants, and the rest are microbes. And the microbe that we're most interested in, as I said, are these mycorrhizal fungi. And they're great. I mean, they're such a bitch to work with, but they're an amazingly complex and fun system. And as I said, it's quite ancient, it's ubiquitous, so 90% of all plant species have interactions with these fungi, right? So this is huge, the huge majority of the plant species out there form some sort of relationship with these fungi. So ancient, ubiquitous. They form these networks, and that's what we've become really interested in recently, that connect different plant individuals underground, right? So already my heart is racing telling about it, right? This is exciting. You've got this microbe, it's connecting different individuals of plants. There's some evidence, and again, I think that also needs to be more evolutionarily informed. But that as the research is suggested now, it sounds very directional and sort of harmonious with organisms that are connected through the network actually helping each other by warning them about predators. There's that kind of stuff out there. But it's definitely true that these networks are used as a conduit for communication. Personally, I think that it's all about eavesdropping and not about signaling, where one is actually trying to help the other. I think it's about plants eavesdropping on each other using the network. But we do know that it's used as a conduit. So the basic is that you're a fungus, you're gathering phosphorus from the soil, you're trading that for sugars from the host plant. And some of the work that we've done has shown that you can discriminate among partners. So just to give you an example of some of the microbial strategies, imagine you have a root system, you've got this fungus, it's intracellular, right? So it can penetrate into the cells. And one of the things that they do is that they actually, once they're inside the host, there's evidence that they can restrict the direct uptake mechanism of the plants themselves. Right, so they're getting in there, and then they're somehow regulating the plant genes themselves and shutting down their ability to take up resources directly. And what that does is it creates...
Can you picture there what's a typical length?
Yeah, no, this is just you'll see. Right now it's just a cartoon, if you will. And what this does is it creates a force dependency. Now there's other examples, really nice work by Martin Hale et al. Shows that, force dependency is something that evolves in mutualisms more generally. They've done work on trees where we see that the tree is providing a protein that actually alters the digestion of the ant so that it becomes more dependent on the tree. Again, this is a case of force dependency. The Dutch, where I'm coming from, they have some of the oldest examples of forced dependencies when they would go in and eradicate all the spice, the East Indies spice trees from countries so that all of the spice trade had to come through them. So this is the forced dependency is a strategy humans use, plants use, and now we see that microbes can also manipulate these types of functions. Okay, so if we know that they're shrewd traders, what we want to do is sort of understand the rules that govern trade in these networks. So I want to show you a short video of how we think it works based on the paper that I sent around that we published in 2011 in Science, where we show that the two partners can discriminate among each other. And this basically shows you how it works. So here's a root, and these red dots are the sugars inside the root. Again, this is a cartoon, not to scale. Here's a spore that's germinating. Now what's interesting is that we've got lots of different genotypes of these fungi in the soil, right? And they're all germinating and making these connections with the root. And actually, if they don't find a host, they actually retreat back into their spore. But if they do make a connection, and here's an example of a successful trade, where you've got the carbon being exchanged for the phosphorus. But here you see there's a certain amount being exchanged for a certain amount. If the root is simultaneously connected to a fungi that is not providing that level of phosphorus, then the trade doesn't happen. So somehow, when the host plant is connected to these multiple strains, it's able to discriminate and give more of the sugars to the fungus that's providing more phosphorus. What's even more interesting is that it can happen from the reverse side as well, where the fungus itself can discriminate among hosts providing different amounts of carbon. So again, when we do experiments where we manipulate levels of cooperation, we see that the fungus tends to associate and trade phosphorus with the host that's providing more carbon. This is basically what it looks like underground and what we're trying to figure out how it works. Okay, so these are the key, and we use a lot of what are called root organ cultures. Have you ever seen these before? So it's a plant with no photosynthetic top. So you're basically just growing a root culture in a petri plate and then inoculating it with a fungal network and studying it from there. And so here, for instance, what you're seeing is just an arena where there's no root, but the two networks of a single network, for instance, could connect these two root systems. So what we found and what we've been arguing for the last couple of years, which is also causing some controversy, is that a market mechanism that is defined in the most simple way, that's competition between partners over the price at which they provide goods, could be the outcome of natural selection. Now, this type of biological market theory has become really controversial, and there's been lots of debate about how it's best used. And I don't want to go into it too much today because I really want to show you some new data. But I just want to make a point because you probably are going to see more of this debate sort of coming down. And biological market theory, it's potentially, it's borrowing from economics. But the key is that the goal is not to draw analogies to human markets. But rather, we can use this type of tool to predict exchange patterns. Now, I think one of the things that we have a problem as ecologists is that we see certain relationships and then we think, okay, context alters it, and so it's different. But we don't really have the tools yet to say, okay, so context alters it, and then we can predict how that interaction will respond. And so I think the power of market theory is that it creates testable predictions, right? And they're very simple. It's sort of the most basic sort of economics, but it does predict, it does allow for us to see these sort of testable predictions about resource exchange patterns. And this is the key part, how they vary across space and time. Right? That's the powers that you can put in different variables into the model and then predict over space and time. And that's really what we're interested in as a group, is looking at strategies and how they change over space and time as context changes. The problem is that we're really limited in our tools. We're really limited in our tool set to study these exchange patterns. It's really nice to go out and study, for instance, this obligate plant mutualism, where you can see the ants, you can see the sort of the resource exchange, you can manipulate the partners quite well. And these types of microbial mutualisms, we can't visualize trade, right? And that's makes it difficult to study resource exchange patterns over space and time. So in the last sort of, I guess, 3 years, we've been developing this technology to try to visualize resource exchange. And what we've done is we use what are called quantum dots, right? So has anybody heard that they use them a lot in sort of medical sciences for imaging purposes of nutrients, but basically we're trying to develop this system where we can track nutrients over space and time. So what they are, they're basically semiconducting nanoparticles. And these nanoparticles, they fluoresce at different wavelengths when hit with a UV source. But the beauty is that they fluoresce in different colors, depending on what is at your core. And so they make these very bright, pure, precise colors. And we can then use those to tag nutrients and finally visually monitor nutrient fluxes in these fungal networks in real time. And it also allows us to sort of quantitatively determine rates of exchange. And this is what we haven't had so much. I mean, there have been radioactive isotopes and things like that, but again, it's been very hard to do different time points and do it visually in real time. Usually you add something and then you see it on the other end. But for instance, this is called RIX, this raster image correlation spectrophotometry, where you actually can quantify the amount of fluorescence in very specific structures in the fungal network. So we can look at different parts of the fungal network and see. So here we're just going to speed through this. These are what the quantum dots look like. We use it with apatite, which is a rock form of phosphorus. Here's some pictures of some plants so we use it in root organ cultures, and then the roots light up in different colors. But you can also use it in whole plants and actually put it in the soil and then you can see it accumulating, for instance, in the petiole in different colors when you hit it with a UV source. Here's a picture of the apatite that's actually as it's going into the hyphal network. And in this picture, it's quite hard to see, but this is actually a different color quantum dot that was coming from the other side. Okay, so what this allows us to do is for the first time we can try to understand the trade strategy of the microbes themselves and where they're sending this nutrient that they've collected from the environment, what they're actually doing with it. So one of the first experiments, sort of the simple one, is just to take a fungus, connect it to 2 different hosts, and just track the phosphorus trade between 2 hosts. But over time, right, because we can use these different colors, we can attach the quantum dots to different phosphorus and then add them at different time points. So we have basically a timestamp of different colors going into the fungal network. So here's just a really simple setup where we have an established root grown under control conditions here on the left. We've got a young root that's connected by this hyphal network. And then what we do to ask sort of an interesting evolutionary question is that we grow the young roots either in just normal control conditions or we make them P-limited. And then we ask, okay, is the fungus going to send more phosphorus to this P-limited root system? Okay, and I actually went... So the fungus is the white lines, is the hyphal network, exactly. Okay, and I'm going to move really quickly through this data, but we can talk more. So these are just to give you an idea of sort of the scale or what kind of data we're getting. This is quantum dot phosphorus per milligram of root. This is the different colors that actually make it into the host root. On the x-axis, you have days since quantum dot phosphorus injection. And it's really basic. We're adding the quantum dot to the established root, so we expect higher uptake in the actual root compartment. And we see that, but we also see a high amount of transfer to the young root as well. And what's interesting from these data, and actually shows it better on the next slide, is that you see this pattern in the control whereby the young root is always getting slightly less, but then in the low P, this changes direction. So basically, you see by the last injection that this low P root starts to get a higher ratio of phosphorus than the control root. So this is sort of the first evidence that these fungus, okay, so they are transferring more phosphorus to the root growing in low P conditions. But why, right? And now this is the part where we're still struggling is, oh, actually, this is also really neat. What you can do with this is then you're thinking, okay, well, it's giving off light. How can you tell if the phosphorus is still in the fungus? Imagine you take a root and you're quantifying the fluorescence. The phosphorus could still be in the fungus, or it could be transferred to the root. But we can use these confocal microscopy approaches to actually zoom in and look at biovolumes and see, okay, what's retained in the hyphae and what's transferred into the root. And here you can see in the young root, under low P conditions, we have more transferred to the root, right? It's just another way of looking at it. But what we're struggling with is trying to measure fungal benefit, right? So we see this fungal strategy of allocating more P to the young root. You would think it would only do that if it receives a direct fitness benefit from this particular strategy. But quantifying fungal fitness is really difficult. So, here we're just looking at extra radical biomass. We can look at spore number, but that's also not really a great measure. But we see the right pattern anyway. It's going in the right direction. That the fungus benefited. It had a higher radical hyphal length. That's just the network that's growing out there. It has a higher amount in low P, but only in the young roots where there was no phosphorus directly available. So we see this sort of evidence of the fungus benefiting, but it's still not satisfying enough to say, okay, this is a strategy, we see it, and it benefits from this strategy. So that's sort of our weak point. Okay, so just to finish up, I just want to show you one more thing. And this is about the rules that govern trade, right? So that was just a really simple experiment about a fungus and a root and where it would go. But more generally, we're really interested in how microbes sense their immediate environment. Can they integrate information across their networks? And does this result in better trade deals? So I'm just going to show you two data slides from this last experiment that we're writing up, and still a lot to do. But what we did here was we took a root system and we used this triple plate compartment where you've got the root system up in the top part, and then you've got the fungal network that spreads out in the other two fungal compartments, yeah? And then what we did was we varied resource distribution across the network itself. So we created 3 treatments of sort of where there was low inequality of the resources across the fungal network, meaning it was 50-50 on both sides of the network. We also did it where it was 70-30 proportions, or where there was really high inequality in terms of the amount of resources that the fungus was exposed to on different parts of the network. And that's what we call our high inequality. Now, what we had expected was that a fungus growing in really high resources, that half of the network, would just benefit and send those resources to the host plant and grow bigger. But instead, we found something very, very different.